Good afternoon. My name is Ben Dwyer. I'm a Senior Research Fellow at Curtin University in Perth, Australia. And today I'll be talking to you about our AMMF funded study, TWIC Fn14 signalling promotes cholangiocarcinoma niche formation and progression. So a number of alterations occur in cancer cells themselves which drive cholangiocarcinoma tumorigenicity. However, there's been increasing focus on the tumor niche in cholangiocarcinoma, which consists of the supporting microenvironment of cells and molecules which drive tumorigenic characteristics. Interactions between tumor cells and the niche are especially important in disease progression, metastasis, and therapy resistance. We're interested in an important immune cell type of this niche, the macrophage. So tumor-associated macrophages, or TAMs, are one of the most abundant immune cell types in cholangiocarcinoma. And they're significant because the presence of a specific type of macrophage is associated with worse patient outcomes. There is a requirement for TAMs to support tumor growth and survival, and Luke Bolter showed that this is mediated by macrophage-secreted Wnt ligands that signal to tumor cells. And he showed by ablating macrophages using liposomal clodronate in a rat model of cholangiocarcinogenesis that cholangiocarcinomas were ablated. There's also an active patterning of tumor-associated macrophages by cholangiocarcinoma cells. So cholangiocarcinoma cells actively secrete molecules which convert macrophages into a TAM phenotype, which express CD206 and also other molecules which promote the tumorigenic potential of cholangiocarcinoma cells. So, given the importance of macrophage-derived signals, we wondered whether other macrophage signals, such as a cytokine called TWIC, could be important in cholangiocarcinoma development. TWIC, or TNF-like weak inducer of apoptosis, is a cytokine that binds to its receptor Fn14 on the surface of cells. After binding, this initiates a signaling cascade called NF-kappa B. NF-kappa B translocates to the nucleus, where it upregulates signals involved in proliferation and inflammation, among others. In the liver, the Fn14 receptor is expressed by bile duct cells and also a mesenchymal cell type stellate cells. During liver injury, there is an inflammatory response. This leads to the influx of monocytes, which are a macrophage precursor cell, and macrophages. And these cells secrete TWIC, which then binds to Fn14 on the surface of bile ducts and stellate cells to promote their proliferation. And bile ducts initiate regeneration in the liver, and stellate cells secrete matrix proteins, which drive fibrosis during liver injury. There are similarities between cells in liver injury and cancer, and because of the similarities of these cell types and their actions, cholangiocarcinoma cells, which are a biliary epithelial tumor, cancer-associated fibroblasts, which are mesenchymal cells similar to hepatic stellate cells, and tumor-associated macrophages. We wanted to know if components of the TWIC Fn14 pathway were expressed in these cell types and whether they had any similar functions in promoting proliferation of fibrosis, which is a feature of cholangiocarcinoma. To assess this, we looked at the distribution of Fn14 in a cohort of patient cholangiocarcinoma cells and found that Fn14 was upregulated in tumor tissue compared to non-involved surrounding liver tissue. We corroborated this data by analyzing publicly available RNA sequencing data, which also showed that Fn14 and TWIC were more highly expressed in tumor tissue compared to non-involved surrounding liver tissue. We then looked at the cell type distribution of these components. Firstly, we assessed Fn14, which is shown in red in cholangiocarcinoma cells, which are pan-CK positive, and cancer-associated fibroblasts, which express alpha-smooth muscle action, in a large number of cholangiocarcinomas. And we found that a significant proportion of both tumor cells and cancer-associated fibroblasts express Fn14. We corroborated this data by reanalyzing a published single-cell RNA sequencing cholangiocarcinoma dataset and confirmed that Fn14 was expressed mainly by cancer-associated fibroblasts and tumor epithelial cells. And we found that the main source of TWIC in cholangiocarcinoma was the tumor-associated macrophages. Having seen that TWIC Fn14 was upregulated in human cholangiocarcinoma, we wanted to know the timing of pathway upregulation, so we utilized a rat model of tumor development. In injured liver, Fn14 was transiently upregulated in injured hepatocytes. However, during tumor development, Fn14, shown in green here, was highly upregulated on tumor cells. We characterized these cells further and found that Fn14 was upregulated on dysplastic biliary lesions, which is a lesion that occurs early in the process of tumor development, suggesting that this pathway upregulation was an event early in cholangiocarcinogenesis. This was mirrored at the transcriptional level, where we saw biphasic upregulation of Fn14 and a gradual increase of tweaked mRNA during cholangiocarcinoma formation. So to summarize, we found that the tweaked Fn14 pathway was upregulated in cholangiocarcinoma on cell subsets that were analogous to those seen in liver injury, Fn14 on tumor cells and cancer-associated fibroblasts, and tweaked on tumor-associated macrophages. The next thing we wanted to find out, given that these pathway elements were expressed in cholangiocarcinoma, was what was the function of these cells. To do this, we utilised 4 well-characterised human cholangiocarcinoma cell lines. We first assessed whether tweak initiated NF-kappa B pathway activation in these cells that were treated with tweak. And we looked at NF-kappa B protein expression, which is shown in yellow. And upon stimulating with tweak, that NF-kappa B translocated to the nucleus, shown in blue. So this shows that tweak was having an effect on pathway activation in these cells. We looked at proliferation in response to tweak in these cell lines, and we found a mixed response depending on the cell type. And this suggested to us that the effect of tweak was perhaps more complex. So we looked at the expression of some pro-inflammatory cytokines and chemokines that were controlled by NF-kappa B. And we showed upregulation of many of these in response to tweak in these cell lines. We also looked at protein secretion into the medium of some of these. The most highly expressed of these was the monocyte chemoattractant protein 1, or MCP1, which is an important molecule in bringing macrophages into the niche, which we'll talk about later. We further assessed the complexity of molecules secreted by cholangiocarcinoma cells in response to tweak using a proteomic screen. As you can see, a lot of the hits we got for proteins that were unique to tweak-treated cells were involved in affecting the niche around these cells, including those involved in immune cell chemotaxis, angiogenesis, and cell activation. Since cholangiocarcinoma cells can affect macrophages, we wondered whether some of these tweak-induced proteins might affect macrophage phenotype. So we wanted to look at this to see whether tweak produced by macrophages increases patterning factors, which drives the conversion of macrophages into a tumor-associated phenotype expressing CD206, and see if there was upregulation of further pro-tumor factors by these tumor-associated macrophages. To do this, we isolated monocytes from the blood of some lab volunteers using antibodies. We differentiated them to macrophages using MCSF and plated these out in dishes. We also took our cholangiocarcinoma cells and treated them with tweak or our control PBS. We let these cells secrete proteins into the medium for 3 days, and then we took this medium that was conditioned by the cholangiocarcinoma cells and put them on the macrophages. And from there, we assessed the phenotype of these macrophages. We looked at marker expression and gene expression. And what we found in our cells that had a high response to tweak was that the conditioned medium from these cells, when put on macrophages, increased the expression of the tumor-associated macrophage marker CD206 in these macrophages. When we looked at gene expression, we saw upregulation of a number of molecules known to be involved in the tumorigenic process. The biggest of these was interleukin-6, which plays a role in cholangiocarcinoma growth. We also saw the upregulation of TNF, which is involved in cholangiocyte overgrowth and pre-malignant transformation, and also MCP1. We also reanalyzed the published single-cell RNA sequencing dataset from cholangiocarcinoma patients and found similar markers to those seen in scar-associated macrophages and liver cirrhosis. One of these upregulated markers was MARCO, which is a scavenger receptor, which is also increased in immunosuppressive tumor-associated macrophages in other tumors. In summary, what we found here was that in response to tweak, cholangiocarcinoma cells produce pro-inflammatory molecules such as MCP1, as well as soluble factors which promote tumor-associated macrophages, which also upregulate pro-tumor factors in MCP1. So we're particularly interested in MCP1 because its role is to signal to bring monocytes that express its receptor, CCR2, in from the circulation into tissues. Monocytes are a precursor for macrophages, so we wanted to further explore if this axis was responsible for building the macrophage niche. To ensure that MCP1 was clinically relevant, we assessed MCP1 staining in our patient samples and found it to be more highly expressed in cholangiocarcinoma tissue versus surrounding liver. We saw it expressed on both tumour cells and stromal cells, and we analysed this further. We looked at a large number of cholangiocarcinoma tissues and looked at expression in pan-CK positive tumour cells, and we found MCP1 overlapped with these tumour cells. We further stratified these tumour cells into Fn14 negative and Fn14 positive subsets, and we found significantly higher expression of MCP1 in Fn14 positive cells, which is suggestive that the tweak Fn14 MCP1 axis is active in human tumour samples. We also looked at MCP1 expression in tumour stromal cells, the CD68 positive macrophages, and alpha smooth muscle actin positive cancer-associated fibroblasts, and we found a proportion of these also expressed MCP1. When we looked at tumour development in our rat model, we also found a similar pattern to Fn14, whereby MCP1 was upregulated on a subset of tumour cells during tumour development. So all of this suggested that MCP1 might be a key molecule in tumour development. We wanted to assess the function of MCP1 in vivo, so what we did was perform a subcutaneous xenograft experiment where we implanted tumours into nude mice and then treated these mice systemically with an antibody to inhibit MCP1. The mice treated with an antibody to inhibit MCP1, we found that xenograft tumours were significantly smaller. When we looked at macrophage populations in these tumours, we found a lower number of both total F480 positive macrophages and also less CE206 positive macrophages. Additionally, we performed a flow cytometry analysis of peripheral blood monocytes, the macrophage precursors, and we looked at the subset of monocytes expressing the MCP1 receptor CCR2. And what we found was that these CCR2 positive monocytes accumulate in the circulation with MCP1 blockade. So this suggested that the mechanism of action of MCP1 is to bring monocytes into the tumour niche, which differentiate into tumour-associated macrophages. And the activation of tweak Fn14, which directly increases tumour MCP1 and indirectly increases macrophage-derived MCP1, may exacerbate this. In summary, tweak Fn14 functions to both establish and pattern a pro-tumour macrophage niche. Having established that tweak Fn14 signalling was significant in cholangiocarcinoma, we wanted to see what would happen if we modulated this pathway in animal models. We used two animal models. We used a dietary model of thioacetamide, and we found that in mice that didn't have the Fn14 receptor, that tumour formation was delayed. And we also used a tail vein injection model. And we saw a really interesting and dramatic change when we overexpressed tweak in this model. And it also modeled a very crucial effect of the niche that we hadn't looked at. So these are whole liver sections of mice with tumours induced by overexpression of the Notch intracellular domain and AKT. And also there's similar mice with tweak overexpression. And as you can see, these tumours become large and cystic, and there's an accumulation of stromal cells around these tumours. We also see a significant increase in the tumour area in these mice. We looked at components of the stroma and found the effect was mostly on the numbers of cancer-associated fibroblasts. We looked at the area of alpha smooth muscle actin staining, which marks cancer-associated fibroblasts, and we found this to be significantly increased with tweak overexpression. We also looked at collagen deposition by these cells using a picrocereus red stain, which marks collagen, and we found a similar increase with tweak overexpression. We then looked further at NF-kappa B pathway activation to see if tweak was activating this pathway. And we used an antibody against a phosphorylated form of P65, which is an NF-kappa B subunit. And what we found was an increased proportion of cancer-associated fibroblasts expressed nuclear phospho-P65 with tweak overexpression. We also looked at a proliferation marker Ki-67. And we saw an increase in the proportion of alpha smooth muscle actin positive cancer-associated fibroblasts that were proliferating. So to summarize, the role of the tweak Fn14 pathway in cholangiocarcinoma is to promote cholangiocarcinoma niche formation and progression. We see expression of the Fn14 receptor both on cholangiocarcinoma cells and cancer-associated fibroblasts. We see the role of tweak Fn14 is to upregulate pro-inflammatory cytokines and chemokines, which build the macrophage niche, primarily by bringing in monocytes to the tumor niche via MCP1, which differentiate into macrophages. And we see a feedback loop whereby tumor-associated macrophages secrete tweak and induce the upregulation of patterning factors from the cholangiocarcinoma cells that promote a tumor-associated macrophage phenotype. We also see the role of tweak on the cancer-associated fibroblasts is to increase the proliferation of these fibroblasts and increase extracellular matrix deposition by these cells. So going back to the start, we know that the interplay of tumor cells with a niche provide a block to effective therapy. And we hope that by targeting tweak and Fn14 might be a way to inhibit this niche formation and lead to the breakdown of that block and increase the effectiveness of therapies. So finally, I'd like to acknowledge all the people involved in this study. Most of the study took place at the University of Edinburgh in the laboratory of Professor Stuart Forbes. This was also in partnership with Curtin University in the laboratory of Associate Professor Nina Turnitz Parker. And we particularly like to thank AMMF for funding this study.